Hello everyone. I am Dr. Cheryl Grace Colasso, professor in the Department of Electrical and Electronics Engineering, St. Joseph Engineering College, Mangalore, India. Recent advances in LED lighting can be used to cultivate crops due to its tunable spectral characteristics, miniature size, and energy efficiency. Energy efficiency of LED lighting for horticulture refers to the ability of the light source to convert electrical energy into radiation that promotes photosynthesis. Photosynthesis is driven primarily by photosynthetically active radiation, known as PAR, which ranges approximately from 400 to 700 nanometers in wavelength, as in Figure 1. However, not all photons in this wavelength range can drive plant photosynthesis equally. For instance, red light, which is in the range of 600 to 700 nanometer, is only 25 to 35% more efficient than blue light, which is in the range of 400 to 500 nanometers. And it is 5 to 30% more than green light, which is in the range of 500 to 600 nanometers, because chlorophyll A and B can better capture red and blue light than green light. Accordingly, light quality affects the plant growth and development via the selective activation of different photoreceptors such as phytochromes, cryptochromes, and phototropin. However, its effects may vary by plant species and plant variety. The wavelength range from 400 to 700 nanometer in the visible spectrum that supports photosynthesis is known as photosynthetic active radiation. Daylight, which extends over the spectral range of 400 to 1000 nanometer, is the most favorable type of illumination for the plant growth. However, it is not always available everywhere and at any time in satisfactory quantity and quality. Hence, artificial light sources have become an interesting factor for producing high quality horticulture products because they optimize the production of phytochemicals through appropriate design of the lighting in combination with controlled timing of the lighting. When it comes to growing the plant indoors, the significance of PPFD, PAR, lumens, and lux might seem confusing at first. As in Figure 2, illuminance and luminous flux is used to measure the light intensity as perceived by humans. PAR is light used by plants for photosynthesis and is measured in PPFD. PPFD is a better predictor of photosynthesis when light sources with different spectral power distributions are considered. Hence, only a fraction of the photons arriving on a plant surface can be used for photosynthesis. Horticulturists measure PAR in plant research. Cultivators try to find the optimum light setting to ensure that their crops produce to the best of their ability. Therefore, it's essential to understand the differences between these light measurements. Figure 3 shows the experimental setup in our laboratory for testing the growth of Malabar spinach in grow chambers under white light, red light, blue light, and natural lighting conditions. There are a large number of LED manufacturers such as Osram, Cree, etc., and many horticultural luminaires available on the market still use monochromatic or a mix of two wavelengths to create the commonly recognized magenta color, which is a mixed spectra of blue and red light with a 1:1 ratio. The use of this type is selected for the growth of leafy green vegetable plants. With the emergence of multi-channel LED-based lighting systems, spectral tuning is now realizable as opposed to the traditional lights. The custom-made spectral characteristics can be obtained by mixing SPDs of LEDs with different dominant wavelengths in PAR region and powering them independently. Figure 5 shows the SPD of one horticulture light available in the market measured with an integrating sphere. This SPD is taken as the target or reference curve to develop our own custom-made spectrally tunable LED source to match the plant absorption spectrum. High-power LEDs, both general and horticulture type, are used in a work due to the high luminous efficiency. The significance of a spectrally tunable LED system is that the spectral power distribution can be tuned to match the plant absorption spectrum. The minimalistic design of a multi-channel tunable LED system has two major considerations. One, use a Gaussian model and a multi-objective optimizing procedure to minimize the error between the simulated SPD curve and the reference or the target curve. And also, this presents a methodology for the optimal selection of the LEDs to achieve the desired tunability. So, these are the contents which I will be discussing in the forthcoming slides. The spectral power estimation for spectrally tunable LED system is estimated using all the technical information available on data sheets of the LED manufacturers, such as full width half maximum, peak wavelength, ppf, total radiant power, and luminous flux. So, and these are the parameters which we have considered for our LED luminaire design. As discussed in the previous slide, initially, the SPD of the LED light source, as shown in figure 5, was considered as the target spectrum. To imitate the target spectrum, the selection of optimal LEDs is required. General lighting LED and horticulture LED database is created in the spectral range of 400 nanometer to 700 nanometer. Figure 6 shows the SPD of 60 LEDs with different peak wavelengths in the database. The range 400 nanometer to 500 nanometers is covered with general and horticulture LEDs. The range 540 nanometer to 620 nanometer is covered with white and phosphor-coated LEDs. The LEDs are modeled using the Gaussian model to get normalized spectrum, which will be explained in the next slide. A small note on FWHM, that is full width half maximum. For radiation with the Gaussian distribution, the width of spectral band is defined as the length of the radiation range, with the emission power is half of the maximum power, as can be observed in figure 7. In the literature, this length is known as the FWHM. So, by utilizing LEDs with different SPDs and controlling the radiometric output of each type of LED, it is possible to make a spectrally tunable LED system capable to produce similar SPD of a specific target with high fidelity. The quality of SPD simulation is intimately linked with the appropriate choice of LEDs. The optimal choice of the quantity and type of LED for the tunable LED system is to simulate reference SPD known as a multi-objective problem. The main idea here is to minimize the quantity of LEDs and at the same time maximize the spectral matching capability. Photosynthetic photon flux. In the process of plant photosynthesis, they absorb light in the form of a photon. ppf is a measure of the number of photons emitted by the light source, and the equation is shown in 1. Luminous flux. Presently, the horticulture LEDs in the entire photosynthetic active radiation are unavailable. Hence, the general lighting LEDs are taken to fill the gap. To match the LED sources, spectral power density and luminous flux, we need to determine the total luminous flux of individual LEDs. The total luminous flux is calculated using equation 2. Photosynthetic photon efficacy defines the efficiency of a horticulture lighting system to convert electrical energy to PAR photons. Higher the PPE value, efficient is the lighting system. And the PP is calculated using equation 3. The modeling was initially performed by calculating the normalized SPD for each LED, S lambda, using the Gaussian model shown in the equation 4 and 5. However, some of the LED datasheet contain the radiant power information or luminous flux or PPF. For these LEDs, the conversion factor F conversion is calculated. This normalized SPD of equation 5 is multiplied with the conversion factor to obtain the simulated SPD as shown in the equation below. For radiant power information, you have to use equation 6, and for luminous flux information, equation 7 has to be used. After modeling of the LEDs and defining the target SPD to be simulated, a simulator was to be implemented. The simulator is designed to have the ability to imitate the target spectrum generated by the LEDs with different power output. The choice of the LED quantity and type of the LEDs was initially tried with LM algorithm. To solve the multi-objective problem present in this work, two objectives were defined. One, minimization of the total number of LEDs, and minimization of the fitting error. So this method determines the power per LED to achieve the target spectrum. So the equation 8 represents the simulated spectrum. The total error is calculated as the average of root mean square of all the individual errors. Ts of lambda is the SPD of the target spectrum and S of lambda is the simulated spectrum. Here LM method is used as an optimization method due to its ease of application in searching the local minimum of a function and for a faster convergence. Figure 8 shows the fitting achieved with the LM algorithm to match the target spectrum. That validates the performance of the algorithm. The selected LEDs was designed to produce a PPF in the range of 30 micromoles per second to 170 micromoles per second and PP greater than 2 micromole per watt, suitable for the plant cultivation. So in conclusion, the primary technical impediment of LEDs for horticultural lighting is the low light output of some current LEDs in wavebands of interest to horticulturists. Although many wavelengths are available, only a limited number are available in very high output configurations. Hence, to increase the yield of the crop, lighting conditions that is suitable for the plant has to be designed. In this presentation, we discussed a custom-made LED light source that was modeled and developed for Malabar spinach cultivation. A methodological approach was discussed to design a spectrally tunable LED system to match the plant absorption spectrum. The study on the effect of LED light conditions on plant metabolites production and biomass accumulation is in process and hence not presented here. I thank